Hello, students. My name is Richard Gomez. I'm 17 years old and I'm from San Diego, California. I'm part of Project Octoscope and I'll be here to teach you for 4 weeks about cells and systems and kind of reviewing what you learned over the week and teaching you new stuff. So this week we'll be starting with Unit 1, Cells and Systems, and we'll go over this week's topic, which is the review of cells. So as a warm-up, I want you to take 2 minutes to write one thing you learned about cells this week. Teachers, feel free to pause the video for 2 minutes in order to let the students write what they learned. So now that you wrote down what you learned this week, let's go through what we did learn. So, first topic was cells. And some important questions that you should know are, what are cells most similar to? What are the two types of cells? And then next you learned about cell organelles. And some questions you need to know are, what do each of the organelles in a cell do? Which organelles are different in a plant cell when compared to an animal cell? Then you went on to learn about plant cells. What makes a plant green? What organelles are specific to animal cells or specific to plant cells? And then you learned about plant versus animal cells. So how would you identify a plant versus an animal cell? And how are plant and animal cells different? So let's start with some notes. Pay attention to the slides and fill in the blanks with relevant information. So if there's a blank, just fill it in with the word or phrase that goes with the blank. So cell overview. What are cells most similar to? Well, cells are similar to a machine with many different parts. Each organelle has a role within the cell, similar to how a machine has a role to keep the machine running smoothly. And on the side here, we have a diagram, right? So up here we have a machine, and each part of the machine does something different in order to let the machine, work efficiently. And it's very similar in a cell. Each organelle has a different role in the cell, and that helps keep the cell healthy and keep it running smoothly. So machines and cells, they're very similar, and that's an easy way to compare cells, So what are the two types of cells? Well, you have prokaryotic. Prokaryotic lack a nucleus, and they're very simple. Right here at the bottom, we have a prokaryotic cell, and you see it's very simple. The DNA is actually, instead of in the nucleus, it's just floating around in the cytoplasm. So prokaryotic cells are very, very simple. And most cells, or a lot of cells, are eukaryotic, and this includes the plant and animal cells. So this contains a nucleus and various organelles. So you see here, in the animal cell, you have the nucleus and various organelles. So that's the difference between prokaryotic and eukaryotic. So let's review the cell organelles and what each one does. You might want to pay extra close attention to these because we'll be doing an activity later that deals with the importance of each organelle. So let's start with the nucleus. The nucleus is the central organelle that controls all the functions in the cell and contains DNA. So DNA is important for a cell, and the nucleus controls all the other functions. So it kind of is the leader, or the thing that keeps everything running in the cell. The ribosomes create proteins in the cell. So proteins are key for cells and for organisms. And you have the endoplasmic reticulum. The endoplasmic reticulum collects and modifies protein and also creates the cell membrane, which we'll talk about a little bit later. But make sure you know that the endoplasmic reticulum collects and modifies protein. So if we're thinking of an analogy, it's like the post office. It kind of collects packages and sends them off to other places. And the mitochondria is the powerhouse of the cell. It creates energy for the cell through cellular respiration. So the mitochondria, creates power. It's like the power plant of the cell. And then the Golgi apparatus further modifies proteins and sends them to a different part of the cell. So kind of different than the endoplasmic reticulum. The endoplasmic reticulum more is you're collecting the packages, and then the Golgi apparatus will be the one that's actually sending them off to wherever it's necessary. And then the lysosomes assist with digestion in the cell and break down nutrients and old organelles. So if something's not working, or if it's just an old organelle that isn't functioning anymore, the lysosome will help break it down and convert it into something that the cell can actually use. And then we have cytoplasm. Cytoplasm is the jelly-like liquid that's contained within a cell and houses the organelles. So it's just where all the organelles kind of live in this jelly-like liquid that's within the cell. And then the vacuole contains fluids, ions, and other important molecules. It's very large in plant cells and helps remove waste products within plant cells. So that's a vacuole. And then chloroplasts, which is specific to plant, help during photosynthesis where plants convert energy from the sun into sugars for nutrients. Finally, we have, second to last, we have cell membrane. It surrounds the cell and acts like a gate, letting some substances in and blocking others. So if it's nutrients or something that the cell needs, it would let it in, but others it would block out. So it's a gate. It doesn't let everything in and it doesn't keep everything out. And then the cell wall, it's specific to plants, and it's the tough outer layer of a plant cell that gives plant cells strength and makes sure they maintain their shape. And it's comprised of cellulose. So let's go back a little bit where we talked about the mitochondria that produces energy through cellular respiration and the chloroplasts, which help produce energy through photosynthesis. These are the equations for cellular respiration and photosynthesis. So you have carbon dioxide plus water plus sunlight results in glucose and oxygen. So that's plants. So plants take carbon dioxide from the air and water from the ground, or if you have a plant, you would water it, and then it converts that to glucose and oxygen. Oxygen is what we breathe. And then on the flip side, cellular respiration, we have glucose. So that's glucose. We can get that from a lot of vegetables or food. And then oxygen, which we breathe in, and then it's converted to CO2, carbon dioxide, water, and ATP. So carbon dioxide, we breathe that out. Water, we urinate that, and then ATP, that's energy, and we use that to move around, and that's basically it. So then we're going to compare and contrast. So here, like I said, the vacuole, you can tell that it's much, much bigger in a plant cell versus the animal cell here. It's very tiny in the animal cell, whereas here in the plant cell, it's much, much bigger. And then we have the cell wall. You can tell that the plant cell has a very rigid shape compared to the animal cell, and that's because of the cell wall. So that's kind of the specifics. And then, of course, you have chloroplasts here, and then that would be specific to the plant cell. So you can tell they have very similar qualities, but of course, plant cells and animal cells have distinct things that make them different from each other. So here we have the cell wall, and here we don't have the cell wall. And when you're looking at things under the microscope, that's actually the most important thing, most important difference between the two. Here, these are examples of plant cells under a microscope, which we'll talk about a little bit later. And you can tell that they have a very rigid shape versus these animal cells that they're more circular, they have a less rigid shape. So that's how you can easily tell the difference between a plant cell and an animal cell if you're looking at them under the microscope. So now we're going to do an activity where we're going to talk about the most valuable organelle. So pick which organelle is the most important out of all of the ones we learned. So this could be the endoplasmic reticulum, the vacuole, whatever you think is the most important organelle. Don't pick the nucleus, because we know that the nucleus is important because it controls all the other organelles. Try picking other important organelles. So don't pick the nucleus, pick something else. So for the first 5 minutes, you'll individually pick which organelle is the most important and write a few sentences on why you believe this is true. And then include its role in the cell and important facts about it. So, if just include its role and then kind of write it out and say why you believe it's the most important. And then for the last 10 minutes, teachers, you will go through the list of organelles and let students raise their hand if they've picked that organelle as the most valuable organelle. So you'll say, okay, students, who picked the vacuole? And some students will raise their hand if they picked vacuole as the most important. So then you'll tally up the totals for each organelle and present them to the class. So you'll say, okay, 10 people picked endoplasmic reticulum and 5 people picked mitochondria, and you can tally it up however you want to. Just make sure students can easily compare and see which one they picked most often and which one they picked less often. And then finally, have a discussion where students present their own point of view, and then students, you will write down 2 other perspectives that you heard from your classmates. So if you picked the mitochondria, but someone said, I think the vacuole is the most important, well, write that down and explain why they believe that the vacuole is important. So you can have different perspectives and you'll be more open. So teachers, take some time to pause the video for about 15 minutes while you do the activity, and then come back and we'll talk about the rest. So now that you've done the most valuable organelle activity and you've discussed with your classmates and you've kind of learned the organelles and reviewed them, now we're going to talk about looking at cells under a microscope. We talked about it before, and we looked at the photos, how the plant cells look different from animal cells. So now we're going to predict, how is it possible to see and study cells? What are the materials necessary to view cells? So if you look at your hand or you look at a plant, you don't see any cells. But how is it possible to get those pictures of cells? So I want you to take 5 minutes to write out a procedure that you believe can be used to view cells. With a procedure, you want to list out the materials. So you want to say, oh, I'm going to need a piece of a plant to look at it. And then whatever you believe, maybe you said you needed, different things, like I need a magnifying glass, I need something. So just list out what you think is necessary to view a cell, and then write out a procedure. So step by step, say, first I will do this, then I'll do this, and then I'll be able to see cells. So take 5 minutes to write it out. It doesn't have to be perfect. We'll go over it a little bit in a little bit. So teachers, take 5 minutes to pause it and let students write their predictions. Okay, well, now that you've predicted how we can see cells, we're going to watch a video that will explain one way to look at cells, and then I'll explain one way that we would look at cells if we were in a classroom and we would be able to do an experiment together. So first, let's watch this video.
Preparing onion microscope slides. You will need the following materials: a microscope camera, microscope slides, a kitchen knife, toothpicks, pipettes, cover slips, iodine tincture, an onion, and a cutting board. Preparing the slides. First, wash and peel the onion. Then, slice the onion as thin as possible. I found that using the thin layer of skin around the onion worked best. Clean your microscope slide with an alcohol wipe. Do the same with a cover slip. Place your pieces of onion onto the slide and reposition them with a toothpick. Add a drop of iodine onto the onion. Drop on the cover slip. Apply pressure to the cover slip, but not enough to break it. Now your slide is complete. Now observe under a microscope. You will be able to see the cell wall and the nuclei of the onion cells. The onion's rigid cell walls allow the cells to be viewed under a microscope, and the iodine dyes the nucleus red, allowing it to be seen. Thanks for watching. Subscribe to see more.
So that video kind of explains one of the processes to be able to see, plant cells under a microscope. So you have to have the microscope, you have to slice it thin so you can see it very carefully, so you can see a thin layer of the onion, and then you'll be able to see it. So one way that we would do this, if we were together in a class and we would be able to do these activities together, would be using foldscopes. So foldscopes are an affordable origami microscope that makes utilizing a microscope accessible for anyone. So these microscopes are very affordable and you can take them almost anywhere. And foldscopes are made of paper and have a small lens that help magnify whatever you're looking at. And foldscopes are actually the inspiration for Project Aquascope. So foldscopes are really important. So when you don't have one of these big microscopes shown in the video, you can use a foldscope and you'll still be able to see the details of cells. And yeah, so now we're going to go through a procedure or one of the labs for looking at cells with foldscopes. So this would be for looking at cheek cells. So this would be an animal cell because this would be our own cells. So the supplies necessary would be a foldscope, 3 slides, 2 cover slip stickers, a paper towel, a toothpick, a pipette, and water. So you'd start out by using the flat end of the toothpick, gently scrape the inside of your cheek. And gently, you want to look at cheek cells, not blood cells. So you would start by taking a toothpick and just going inside your mouth and scraping the side of your cheek. And you just want to do it very gently, and then that's the first step. So then you will place a cover slip on the paper side of one of the slides. So you'd place the cover slip on one of the sides, and then you'd put the cheek cell from the toothpick onto the center of the slide. So you just kind of slide it off, and then get what you have and put it on the slide. And then you'd place another cover slip on top of the cells, and then stack the plated side with 2 other blank slides before inserting the stack into the back of the foldscope. It's kind of hard to demonstrate without actual foldscope, but essentially you have a slide similar to the one in the video, and you're preparing it, and you're putting another cover slip on, and then you're putting it into the side of the foldscope, kind of like right here. You would put it onto the side, and you'd slide it in so you'd be able to see it with the lens. And then you observe the cheek cell by focusing in and out of the cell. So the foldscope has a built-in kind of mechanism where you can focus and try to zoom in and out to try to get the best view of the cell. And essentially, that's basically it. You could also use some dye, some kind of like iodine in the video. The iodine, as she said, serves to dye the nucleus so you can see it a little bit better. So if you have some dye, that could also help in seeing the cells. So once you're done, you'll be able to see something like this. This is an exact photo, but this seems to be more of a high-powered microscope, but you'll see something very similar. So here you can see the cytoplasm, the nucleus, you see some of the organelles, and then some bacteria. So, there's naturally bacteria in your mouth, so when you scrape it, you'll be able to see some of it. And that's essentially what you'll get with essentially how you can see cells. So you don't need a magnifying glass or anything. You need a microscope, and then once you have a microscope, you'll be able to zoom in very closely and see these cells. So you can do that either with a big microscope or with a foldscope. So that's basically our lesson today. If you still have time in class, take some time to review the notes from today, or go back and do some of the worksheets from earlier in the week. If you didn't finish them, maybe get some friends to help you out and compare answers. So thank you very much. Good luck and have a great day.